This is a special video for the auditory system, and I'll talk about the computational model of cochlea. A computational model in neuroscience is a means of understanding and studying complex system, which is the brain, using an algorithmic or mechanistic approach. Nowadays, many scientists are modeling human perceptions. Modeling the cochlea is helpful to understand one of the most important human perception, hearing. Today, I will talk about the Cochlea modeling based on the recent paper. Deep neural network models of sound localization reveal how perception is adapted to the real-world environment by Francl and McDermott from MIT. They applied a deep neural network with human ears and trained to localize sound in a virtual environment. The resulting model localized accurately in realistic conditions with noise and reverberation. In stimulate experiment, the model exhibits many features of human spatial hearing, but when trained in an unnatural environment without reverberation, noise, or natural sound, the performance characteristics deviate from humans. This is a simple architecture of their work with human auditory procedures. Here, I will only focusing on the Cochlea model and its output, the cochleagram. Before starting, I will give you a brief background explanation about the first part of the human auditory processing. The sound is captured by the external ear and hits the tympanum. Here, the sound wave is transformed into the tympanum's vibrations and transmitted by three small bones, malleus, incus, and stapes. The movement of stapes oscillates the liquid of the cochlea. The oscillation of the liquid causes the movement of the basilar membrane. The shearing movement of the basilar membrane and the tectorial membrane cause the deflection of the hair bundle of the hair cells. Until here, the sound energy is still mechanical information. But from the deflection of the hair bundle, their ion channel is activated by the gating spring mechanism, and this is the transduction from mechanical information to electrical information. So this is the first part of the auditory processing. Now, let's move on to the computational model. We can call Cochlea Model the computational auditory scene analysis, which is a study of auditory scene analysis by computational means. As the first stage of causal processing, the cochleagram creates a time frequency representation of the input signal. By mimicking the components of the outer and middle ear, the signal is broken up into different frequencies that are naturally selected by the cochlea and hair cells. Because of the frequency selectivity of the basilar membrane, the filter bank is used to model the membrane, with each filter associated with a specific point of the basilar membrane. Here, I would like to explain what and why time frequency representation, but first, let's talk about the filter bank or ERB. So, what is the filter bank or ERB? Equivalent rectangular bandwidth gives an approximation to the bandwidth of the filters in human hearing. Also, it is called a rectangular bandpass filter. But why is the rectangular bandpass filter represent human hearing? It is because of the characteristic of the cochlea. In the cochlea, the basilar membrane can distribute specific stimulus energy due to the structure which has different mechanical properties. And the base of the basilar membrane responds to high frequency, and the apex of the basilar membrane responds to low frequency. Since the basilar membrane has a tonotopic arrangement, every cochlear hair cell is most sensitive to stimulation at a specific frequency. The resolution of the frequency that the cochlea can detect mostly depends on the inner hair cells. The human cochlea contains about 3,500 inner hair cells, and the range of the characteristic frequency is about 20 Hz to 20 kHz. The time frequency representation is the representation of the signal with time and amplitude by time frequency and power. Each frequency represents each hair cell's position on the basal membrane and its characteristic frequency, and the power represents how much they are activated or fires on its characteristic frequency. Lastly, at which time point they were activated. These three informations comes from the cochlea. However, it is not efficient to calculate and apply all 3,500 inner hair cells' characteristic frequencies. Therefore, approximate frequencies range can be calculated by Glassberg and Moore's formula. For example, a human can detect sound in the frequency range from about 20 Hz to 20 kHz. In this case, the number of filters in the filter bank will be 39. Now, by passing filter bank, the outcome is the subband. The subband is encoded by spiral ganglion neurons in the cochlea and sent it to central nervous system or the neural network. The central nervous system can gain information about stimulus frequency in two ways: a place code and a frequency code. A place code, the fibers are arrayed in a tonotopic map in which the position is related to characteristic frequencies. A frequency code is the fibers fires at a rate that signals the frequency of the stimulus. It is particularly important when a sound is loud enough to saturate the neuronal firing rate. Although fibers with different characteristic frequency respond to such a stimulus, each provide information about the stimulus frequency, its firing pattern. In our cochlea, encoding a place code and a frequency code differently is important, but output of the Cochlea model, cochleagram, is already included in both place code and frequency code. Now, how to encode the Cochlea model's response or provide especially the stimulate spatial sound information. In the paper from Francl, they're saying in order to provide fine timing information, we use rectified subbands of the signal instead of the envelope of the subbands. Here is the example of taking subbands envelope or rectified subbands. Let's imagine this 3 Hz sine wave is the response of the cochlea. When you take an envelope of the signal or subband, you can get the general picture of the firing rate. On the other hand, if you take the half-wave rectified signal, you will get more information about the time. Now, let's move on to the cochleagram. As I explained before, the outcome of the Cochlea model is the time frequency representation. I will give you an example provided by Francl. This is the sinusoid signal of 100 Hz with 40 harmonic frequencies. By passing 39 filter banks of the frequency range from 20 Hz to 20 kHz, the outcome of the Cochlea model will be like this. The x-axis and the y-axis represent the time and frequency, and the color bar represent the intensity of the firing rate displayed by the warmness of the color. Lastly, the response of the cochlea or outcome of the Cochlea model transmit to the brain or neural network. Of course, the model of brain, which is the neural network, is another story to understand, but for this video, I hope you all have first step to understand what is the Cochlea model and how it works. I would like to conclude this video by giving the most famous example of the sound localization, the cocktail party sound with stereo recording. These are the two channels from the sound you heard in 3 seconds. and this is the response of the Cochlea model that we can consider, this would be the response of the cochlea. Thank you for watching. See you then.